The last type of transport across the membrane describes transport that uses vesicles in order to move large macromolecules and even solid particulates. Two major types of vesicular transport will be discussed here: endocytosis and exocytosis. Endocytosis encompasses phagocytosis, which means cell eating. Macrophages, a type of white blood cell, are particularly good phagocytic cells. They extend portions of their cell membrane called pseudopods to surround objects in the extracellular fluid, much like the blob would extend portions of its monster self to envelop its victims, with the victims now immersed in the gelatinous blob. Once the phagocytic cell envelops its prey like a bacterium, some of the membrane pinches off, forming a membrane-bound envelope or vesicle around the bacterium. This vesicle is better named a phagosome and will fuse with a lysosome, which is discussed or described later, so that digestive enzymes can degrade the bacterium. In picture B, you see another type of endocytosis called penocytosis. Penocytosis means cell drinking. It is very similar to phagocytosis, but instead of large particulate matter, the cell simply internalizes the extracellular fluid. Literally, it is as if it is sipping or sampling the extracellular fluid, and does so to obtain dissolved nutrients. Receptor-mediated endocytosis is really a chemical signal system. Chemicals, shown here as green spheres in picture C, bind to integral proteins in the cell membrane that function as receptors. Once the receptors are bound to the chemical signal, part of the membrane breaks off and again creates a vesicle. These chemical signals can then alter the activity of the cell. With exocytosis, the vesicles carry waste material, or chemical signals, and the vesicles fuse with the cell membrane and release the contents into the extracellular fluid. Pictures of this process are found within the red circle. The word cytoplasm means cell-forming material, and this is an area of the cell that is internal to the cell membrane, but external to the nucleus. The fluid substance, or intracellular fluid, is jelly-like, and the organelles are scattered within it. This fluid is called cytosol and has water, ions, enzymes, sugars, amino acids, etc. Within the cytosol are proteins that are arranged as rods that span the cell. This network of rod-like proteins gives support to the cell. There are three main types: microfilaments, intermediate filaments, and microtubules. Microfilaments and microtubules are made from globular proteins. The small proteins interact with each other to make a larger strand. Intermediate filaments are made from proteins that are more fiber-like. Microfilaments are the thinnest of the three and are made from the globular protein actin. In muscle, actin filaments interact with myosin for muscle contraction and when the cell divides during cytokinesis. Microtubules are the largest of the three and again are globular spherical proteins called tubulins. They arrange to make a long hollow tube that is stiff but bendable. Microtubules radiate out from centrosomes and anchor the organelles of the cell in place. They are also involved in making the mitotic spindle, which will be discussed shortly. Microtubules are very labile and are constantly assembling and disassembling. Those used in the centrioles to make mitotic spindles are very stable. Intermediate filaments, as their name would suggest, are between the microtubules and microfilaments in size. They are the most stable of the three, and their use in the cell stems from their tensile strength. They resist pulling forces and act like guide wires of a bridge. They are also used to make desmosomes, a type of cell-to-cell attachment that you will learn more about in your tissues chapter. The organelles I will discuss include mitochondria, lysosomes, peroxisomes, the endoplasmic reticulum, ribosomes, the Golgi apparatus, the centrioles, and the nucleus. Mitochondria are bean-shaped organelles, and they are responsible for converting the stored energy in the food we eat into the type of energy the cell can use to do work. This cellular usable type of energy is called ATP, adenosine triphosphate. ATP is used for chemical reactions throughout the cell. Mitochondria are membrane-bound organelles. They have a double lipid bilayer, just like the plasma membrane. However, the internal membrane is unique in that it is highly convoluted. The folds of the internal membrane are known as cristae, and they increase the surface area. And as shown in Part B of the picture, this allows more space to embed important proteins that are collectively known as the electron transport chain. These proteins are responsible for moving electrons, and this process is required in order to make ATP. Oxygen must be present for this process to occur, and the end result is glucose and oxygen are converted into ATP, carbon dioxide, and water in a general process known as aerobic respiration. A neat little fun fact is that mitochondria have their own circular DNA. 1% of the total cellular DNA is found in mitochondria, and the genes found on this DNA are used in the electron transport chain, or for protein synthesis in the mitochondria. The genes on mitochondrial DNA can be mutated, and known genetic diseases stem from damaged mitochondrial DNA. Lysosomes are membrane-bound organelles that contain digestive enzymes within them. Lysosomes fuse with phagosomes to digest the contents. Lysosomes are also needed to digest old organelles. Some white blood cells have a high number of lysosomes and are known for their ability to attack invading cells. Tay-Sachs disease is an inherited condition where the person's lysosomes lack an enzyme called hexosaminidase A that breaks down certain glycolipids in cell membranes. It is an autosomal recessive disease. The glycolipids are particularly abundant in nervous tissue. As the glycolipids accumulate, they become toxic to the cell and interfere with nerve function, resulting in diminishing mental capacity, blindness, loss of movement, and finally, death. Death usually occurs by age 2. It is extremely rare, but can be common in certain isolated populations, like the Ashkenazi Jews. Peroxisomes are very similar to lysosomes in that they are a membrane-bound sac, but instead of being filled with digestive enzymes like the lysosomes, they contain enzymes that are capable of destroying reactive oxygen species and oxidative radicals by ultimately forming hydrogen peroxide, which is then destroyed. They have enzymes like catalase and peroxidases in order to tamper the damaging reactive oxygen species, abbreviated as ROS in the scientific community, and not to be confused with the ever-threatening ROUSs, or rodents of unusual size, from the movie The Princess Bride. Peroxisomes are necessary to help the cell by protecting it from the damage oxygen can do to the cell membranes, proteins, lipids, and DNA. The endoplasmic reticulum is continuous with the outer nuclear envelope. In fact, the outer nuclear envelope is derived from the endoplasmic reticulum after mitosis occurs. The ER is a large expansive system of membrane envelopes, and its name means network within the cytoplasm, and its membranes account for more than half of the membrane surfaces within the cell. There are two types of ER, rough and smooth. The rough ER is at the end of the blue arrow. The rough ER has ribosomes attached to it, giving it a rough or stippled appearance to its surface. This is the site of protein synthesis for proteins specifically targeted to another organelle, an integral protein in the cell membrane, or an extracellular protein. I'll speak more about the rough ER on the next slide. The smooth ER is shown at the end of the red arrow. Instead of envelope-like structures, its membrane branches and forms more of a lattice structure. Ribosomes are not associated with it, and thus protein synthesis does not occur here. Its function is to make or break down lipids, and so it should come as no surprise that smooth ER is abundant in cells that make steroid hormones and in liver cells where lipid-soluble drugs are detoxified. Lastly, the smooth ER is important for storing calcium ions. This will be very important to you during our Unit 2 discussion on muscle contraction. Calcium is a very important cellular trigger. By that I mean when calcium is dumped into the cytosol from the smooth ER, great things will happen. So calcium is your cell's molecular flip switch. When it is released from the smooth ER, the cell changes radically its activity. When it is resequestered into the smooth ER, the cell will go back to its normal activity. As I said in the previous slide, the rough ER has ribosomes attached to it. This is the site of protein synthesis for proteins specifically targeted to another organelle, an integral protein in the cell membrane, or an extracellular protein. How does this happen? Well, the ribosome, shown here as an orange structure that looks like an upside-down hamburger bun, slides along the messenger RNA, or mRNA. As it does so, it scans the nucleotides in the messenger RNA 3 at a time. The three nucleotides collectively are called a codon. This is the process of translation. The sequence of the nucleotides within the codon determines which amino acid will be placed in the growing peptide strand, shown here as a lighter orange strand that is coiling into the cisternae, or cavern, of the rough ER. Translation is discussed in more detail during our lecture called Gene Expression, but this should be review for you from Bio 20. Once the protein synthesis is complete, a portion of the ER buds off with the protein within it, and the protein-filled vesicle will now go to the next organelle in our series, the Golgi Apparatus, which will be discussed shortly. Ribosomes can be found free in the cytoplasm or bound to the rough ER, and when they bind to the rough ER, it makes the ER, the endoplasmic reticulum, have a rough appearance. Therefore, we say rough ER. These organelles are not surrounded by a membrane, and they are made up of a protein and ribosomal RNA. The ribosomal RNA is made by the nucleolus, which I will talk about later. Ribosomes scan across a messenger RNA during a process called translation to make protein. The process of gene expression to make proteins will be discussed during your gene expression module. Free ribosomes will translate messenger RNA to make proteins that will be soluble in the cytoplasm, and ribosomes on the rough ER will make proteins that are either destined for another organelle, to be inserted in the cell membrane, or exported out of the cell. The Golgi apparatus is part of the pathway for a protein destined for another organelle, or a protein destined to be inserted in the plasmalemma or outside of the cell in the matrix. The Golgi apparatus sorts, processes, and packages these proteins. I like to think of it as the postmaster of the cell, in that it routes the proteins to the proper destinations. This membrane-bound organelle consists of a series of disc-shaped envelopes called cisternae, or as your book describes them, they are like hollow saucers, one cupped inside the next. Proteins made by the rough ER move to the cis side of the Golgi apparatus, or the receiving side, and then are processed within the cisternae, and then bud off in vesicles from the trans side. The vesicles may fuse with the plasma membrane in order to secrete the proteins, or be contained within the cell as a lysosome to fuse with a phagosome to degrade and digest the phagosome's contents, or it can simply fuse with the cell membrane to contribute to the plasmalemma, and the proteins become integral proteins. The centrosome is found near the nucleus. It is an organelle, but it is not membrane-bound. The centrosome consists of the centrosome matrix, which is a haze of protein, and a pair of centrioles, which are made up of microtubules that are arranged in 9 groups of 3. These microtubules do not disassemble and are very stable. I like to think of the centrosome as the city of LA, with the centrioles as the skyscrapers, and the smog haze as the haze of proteins called the centrosome matrix. The nucleus, which means little nut, is often thought of as the control center of the cell. Deoxyribonucleic acid, or DNA, is found here. More will be discussed about DNA during the gene expression portion of this module. DNA provides the instructions or genetic blueprint for your proteins. Some cells, such as skeletal muscle, are multinucleated, which means they have more than one nucleus. Most cells are mononucleated, and the most abundant cell in the body, the red blood cell, lacks a nucleus, so we say it is anucleate. The nucleus is surrounded by a double membrane nuclear envelope, with the outer membrane being continuous with the endoplasmic reticulum. Scattered in the nuclear envelope are nuclear pores that allow protein and RNA molecules to pass into and out of the nucleus, respectively. The nucleolus is a specialized part of the nucleus where ribosomal RNA comes from that is essential for translation. This concludes our discussion of the cell and its organelles. In the next module, I discuss the cell cycle.